Hello, thanks for tuning in. I'm Holly Shields for Kalkine Media. Welcome you all to another edition of Executive Corner Expert Talks. Today, I'll be shedding light on Ochre Bio, which develops RNA therapies for chronic liver diseases. Built on decades of leading genomics and drug development experience, the company is situated within the heart of Oxford's biotech innovation hub. And joining us today to share their insights is CEO Jack O'Mara. Jack, thanks very much for making time for us today.
No problem. It's lovely to be here.
It's a pleasure to have you on the show. Now, first and foremost, congratulations on the launch of Ochre Bio's Liver ICU in New York City, which is a world first for human liver research. What are the goals of this new venture?
Yeah, so when we started Ochre, we really were building the company around three challenges that have held back liver disease drug development. The first is that the biology is still very poorly understood for the disease. And the second is that the models we use to test out biology or test out new therapeutic candidates don't typically recapitulate the disease very well, such as animal models or others. And then lastly, clinical trials for chronic liver disease are a real challenge because it is a silent disease, and therefore trials need to be very long and are very expensive, which is a disincentive to small companies. So when we started the company, we were thinking about these challenges and really built it on amassing very rich genomic data to better understand the biology of the disease, and in doing so, using advanced genomics like single cell sequencing and spatial sequencing on human tissues like diseased human livers. But then secondly, we wanted to design models that allowed us to validate that biology. So what we work on are actually discarded donor livers. So livers that have come to a transplant center but can't be used because they are too diseased. We take them in partnership with transplant centers in our new site in New York, maintain them on these new perfusion devices, which effectively keep livers alive outside the body. And we use these otherwise unusable or non-transplantable organs as a means for us to test if our therapies are actually going to be successful when they go into clinical trials. So that second problem is really what New York is focused on. It is for us to validate the RNA medicines our team in Oxford has been working diligently on and make sure they actually work when you deliver them to a diseased donor liver. And I can get to the third problem, or how we're solving some of the clinical trial challenges as well, if it's of interest.
Yeah, absolutely. I'd love to hear the third problem there.
Yeah, so as I mentioned, chronic liver disease trials like NASH, which is the clinical term for fatty liver disease. The challenge is the biomarkers are quite difficult to meet in terms of what the endpoints the regulator sets, and therefore you have to have these very long, very expensive trials. And not that dissimilar to neurodegeneration or Alzheimer's. The endpoint you're looking at doesn't typically give you it's very difficult to get a conclusive readout from that, which has been some of the challenges for other chronic diseases of aging. So for us, our first program, the first product we hope to bring into the market to improve patient lives is actually in the transplant space. So these same category of slightly high risk or slightly diseased donor livers that patients ultimately now who are going for liver transplant typically receive because of such a shortage of donor organs. We hope to deliver therapeutics, RNA medicines like the ones we designed, to improve outcomes for patients who receive this category of slightly fatty, slightly older donor liver, and thereby initially get into the clinic, show our efficacy, show we can impact patient lives, and then stepping stone our way into larger diseases like the NASHs or others of the world. And that way we can, a little bit like incrementally, one small step at a time, build confidence that our medicines are really going to impact the human health at the scale we believe we can.
That's incredible to hear, I must say. It sounds like you really are slowly but surely getting there to your with your work and your expansion, and it's slowly growing, and the addition of that New York body as well is really helping that research come along. So that is ultimately great to hear. Now I understand as well that Ochre Bio is developing therapies for some of the biggest liver health challenges, range rather from increasing donor liver supply to reducing cirrhosis complications through metabolic stress, metabolism, and regeneration programs. Now, how are all these therapies unique?
Yeah, so modality that we're really excited about, and now the world is very excited about it, is RNA medicine. I would like to say we were doing it before it was cool, and since the news of COVID. But RNA medicine is an amazing new way to develop therapeutics for challenging diseases, because it's incredibly, the duration of effect one can have can be 6+ months. So you can get one jab in the arm and that drug could be working for 6 months, even to a year with some of the new breakthroughs. It's also highly safe and highly specific, particularly the type of RNA that we use as a company, which is called an siRNA. So if you think of mRNA as the ability to turn on genes that you want to turn on, siRNA is sort of the opposite effect, where you turn off genes that are potentially having a deleterious effect. So we're very focused on using siRNA, but the benefit is there's a delivery technology, and this is getting a little bit sciencey potentially, but it's essentially a sugar we attach to the end of the siRNA, and it gets soaked up by the liver incredibly quickly and stays in the liver for 6 to 12 months. So we can have this incredible duration of effect for a therapeutic that's not typically possible with small molecules. The other thing, and this is sort of a more nuanced point, but I'll try and explain it as clearly as I can. The other thing that working in RNA allows us to do is it allows us to really speed up our development cycle. So in drug development, typically you pick a target, then you might even take a couple of years to optimize your small molecule to treat it, and then you go into later stages of clinical development. So you've got a very long lead time to go from what is a biological hypothesis. So if I believe a gene is driving a disease, it takes quite a while for me to get to a medicine that will be able to treat it. But with RNA, and Stefan Bancel and some of the folks at Moderna have done an amazing job of articulating this idea, this ability to write medicine, in that once you know the sequence, the story goes that at Moderna it took them 48 hours to get to the medicine, and it should have been faster and so on. But it really is that quick to go from what you believe is a potential biological hypothesis, so a new gene that you've uncovered in using our genomic data or otherwise, to then very quickly transitioning that into an actual medicine that could be used to treat it. So then we can then test it in our New York lab, where we'll see in an actual diseased human liver if our therapies are really having an effect in the way that we think will be really powerful for patients. So that cycle time, that ability to go from therapeutic hypothesis to drug and back again in a preclinical environment, allows us to just really focus on making sure that the biology that we're going after, so the potential gene targets or otherwise, are going to be the most effective for patients. And that, I think, is a real breakthrough, and a real going to be really impactful for human health, we hope.
It does certainly sound like a breakthrough, and you've got a whole lot of advantages there, as you just mentioned. So that's definitely good news to hear. Now, I'd just like to touch on the hidden fatty liver epidemic, which we've heard of quite a bit as a silent killer. It's widespread and often undiagnosed. How can genomic research, in your opinion, help us to really understand it and eventually eliminate it?
So this is a great question, and it's one of the sort of founding hypotheses of the company, is that we do need to really study the disease itself in a way to get to better ideas around how to treat it. So when we first started Ochre Bio, one of the first major projects we kicked off in partnership with Oxford University was to take 1,000 liver biopsies of varying stages of the disease, the silent disease that is so hard to treat. And we did this by working with transplant surgeons who had biopsies of various different stages of patients who'd been in to see them, or organs that have been donated for transplant, and then used single cell and spatial sequencing technologies to really study disease progression across those livers, to give us clues as to what at a genetic or transcriptomic level is changing as the disease gets worse and worse, and potentially through doing that gives us clues as to, wow, if this is really, but we see a lot of correlation between this changing and the disease getting worse, maybe that's a potential way for us to reverse or treat or prevent the progression of that disease. So genomic technologies have this incredible ability to really study genes and cells at the fundamental unit of life, the really the basis of the disease, and then give you interesting ideas around what might be the best way to treat it. And that's, again, going back to I'm very excited about our New York lab, as you can tell, but then the ability to go and study in an actual discarded donor liver, whether your medicines work, just is an incredible opportunity for us to hopefully make a real impact in the liver disease space.
Undoubtedly, it absolutely sounds like an incredible opportunity there. And you've obviously got all this amazing technology on your side and this research and this facilities as well. It's ultimately great to hear. So we do look forward to seeing everything you have coming up. And speaking of which, what else can we look forward to seeing from Ochre Bio for the remainder of 2022?
So at Ochre, internally, we're very focused now on taking all of the research we've been doing since 2019, building these potential therapeutic candidates in Oxford and testing them in high throughput sense in our in this perfusion lab in New York to really get confidence that the drugs that we've been working on are going to have the impact we hope they'll have for patients. So for the rest of this year, it's all about going through that liver perfusion work and ultimately which will inform what drug candidate we hope to take into clinical trial over the coming years, which is going to be a really momentous occasion for the company.
Absolutely, that sounds like really important stuff there, and we'll definitely keep an eye on your progress in the upcoming year and beyond. But with that said, thanks very much for joining us today on Executive Corner Expert Talks and sharing your insights.
Very happy to. I hope I didn't get too nerdy or scientist.
Not at all. It's a pleasure to have you on the show. And thanks for your time as well, viewers. If you've just joined us, that was Jack O'Mara, CEO of Ochre Bio. And if you missed any part of that chat, you can catch the full interview on our YouTube channel at Kalkine Media, and make sure to subscribe. I'm Lo Shields, money to stay apprised and invest wise with Kalkine.